The next presentation will be by Matt Strain from the University of California, San Diego, and the title is Terminal Dilution Assays Reveal Diverse Activation Responses Within the Latent Reservoir.
Okay, thank you. I'd like to thank the organizers also for inviting me. We've been hearing about a lot of different approaches to try to target the latent reservoir, and I'd like to talk to you just a little bit about how to determine whether this has been successful. This has been an issue in clinical studies for decades, and it is usually said that the gold standard assay is the quantitative viral outgrowth assay. This measures definitively cells that can be rekindled to produce infectious virus, and since ultimately the interruption of antiretroviral therapy and rebound after interruption is the definitive test that any assay should try to predict, this is the most direct measure. So the way this works basically is the CD4 cells are activated ex vivo. Those produce virus, which is then propagated typically for a couple of weeks in feeder cells, which various types of feeder cells have been used, but typically they're CD4 from one or two additional patients. This assay is time-consuming, expensive, and the reproducibility has really never been studied all that carefully. The basic way that you get a number out of this assay is by assuming a binary dilution assay structure, single-hit Poisson kinetics, and looking at which dilutions actually allow virus to propagate out. Now, although this is called the gold standard, it is not actually used as frequently in studies as HIV DNA, which is far easier to measure. Unfortunately, these two measures are really quite different from one another. So this was shown, I think, most completely in a study by Erickson et al., which compared a number of different measures of the size of the reservoir, and they found, as others had in smaller studies, that the QVOA and HIV DNA assays did not correlate well. In fact, in 3 of the 4 HIV DNA assays they looked at, there was not a significant correlation. Furthermore, as you can see here, by comparing the identity line with the data in that study, there is more than a 2 order of magnitude difference in the magnitude of the QVOA and HIV DNA, which is now has now been definitively shown at a quantitative level by Ho et al. In this Cell paper to be due, at least in significant part, to the fact that the HIV DNA provirus is defective. Now, in this study, they found that by sequencing, full-length sequencing, or sequencing of overlapping amplicons of essentially a full-length provirus, 88% of the discrepancy, 88% of provirus was defective. That still only explains 1 order of magnitude difference. I told you there's more like 2.5 logs difference between DNA and QVOA. So this is not the whole story. And of the 12% that lack discernible defects, still the majority of those were unable to propagate viral infection. So clearly there is still a lot of room for something in between, or for extrapolation between the HIV DNA assay that is widely used because it's convenient and the QVOA assay that is the most direct measure of what we really care about. So in this study, we were basically interested in trying to fill in some of these gaps. Other people have done similar things recently. There was a publication by the Mellers group that has looked at approaches to using activation ex vivo, as is done in the QVOA assay, but now, rather than propagating the infection, we're going to look at single-round production of HIV RNA. Nicolas Chaumont's lab has also done this with the tilde assay, which you've heard about some earlier today. So we're actually going to look, I'll talk about several different types of HIV RNA here. We looked both intracellularly and also at extracellular produced cell-free virus and at a number of different targets, which turned out to be somewhat more interesting than we had anticipated. So most of the study dealt with a cohort of 16 patients. There were 22 for some portion of it. All of these patients had initiated ART during chronic infection and had been suppressed for at least 1 year. It's actually, as you'll see, a rather unusual group of patients in that the size of the reservoir is unusually narrow, which makes it somewhat more challenging to demonstrate correlations between the assays. But we now have actually added a few acute patients, and it does seem like the trends hold. So in addition to the HIV DNA, which we did by droplet digital PCR as we published, we used a QVOA assay. This is based on the idea published by Shan et al. In 2013 of using a CCR5 feeder cell line rather than using the human CD4 cells to propagate the infection. It has the advantage of being more uniform, but you do have to watch those cells very carefully because they can lose the CCR5 on the cell surface. So you have to monitor them every week for expression levels in order to use this feeder cell line. And then we looked at HIV RNA also by digital PCR, or RT digital PCR, looking at targets I'll go through in a second, and we looked both at cell-free virus in supernatant and at cell-associated RNA within the activated cells. RNA was extracted using magnetic beads with the MagMax kit for the cell-associated RNA. For the cell-free RNA, we used a kit used an approach. It is basically a kit that is not commercially available from Hologic. It's part of their clinical viral load assay, and this uses HIV-specific sequences. There are two of them, one in LTR, one in integrase, that pulls down the viral RNA, and as you can see here, we do get somewhat better recovery using this assay versus the commercially available ones. And this approach has been validated because it's part of their clinical assay for a variety of different subtypes. The targets that we primarily ended up focusing on were GAG and a multiply spliced RNA target that spans the TAT-REV splice junction. These are among the many primer sets described in Schmidt +1 paper in 2010. We did also look at what's been called the poly-A RNA primer set that has been described by the Silicano group that targets the 3' LTR into the poly-A region so that it specifically picks up all HIV transcripts. That gives slightly better sensitivity than the GAG assay, but is somewhat more difficult to interpret and can't be multiplexed. So I'm not actually going to show you those data, but we did do comparison studies. So our hope really in starting this out was that we could just do a single activation, detect RNA, and perhaps leaning to some extent on the precision of the RNA measurement by RT digital PCR, be able to determine a reservoir size just from a single RNA measurement, or perhaps doing this in triplicate initially. However, that turned out not to work too well. There's a lot of stochasticity in this process, and so we found that running a single well by digital PCR gave results that did not correlate well with any of the other assays. And so these are some of the stochasticity data. I'll skip through this. So therefore, in order to get a quantitative result, we went to an approach similar to the QVOA assay, in which we activate the cells in the presence of raltegravir for 3 days, and then collect in a series of wells that have been serially diluted prior to activation RNA both in supernatant and in cells. And then the quantitative number of inducible cells is computed as it would be for the QVOA by assuming a binary assay. So we are getting additional information out here that's not ultimately used in the readout because we have digital PCR data, so we have a copy number produced at each of these levels that is not the readout that I will be showing you, but it does allow us to actually independently look at how much RNA is produced within these cells. And we did a relatively large number of replicates in the hope of being sure whether any associations we saw were due to the size of the assay. Okay, so now in between the QVOA and HIV DNA, we have these RNA assays that we're going to look at. This is an example of the RNA, what the RNA data looks like for a single patient. So we have a series of threefold serial dilutions, 8 dilutions, 12 replicates, and from each well we harvest cell-free RNA from the supernatant and assay it for GAG, which is shown in red here. The shade of red, it tells you how many copies were detected in that well, and then all the ones that have no shade, that we didn't detect any. Within cells, we looked at both the unspliced GAG and the TAT-REV targets, which are green and blue. Now, this patient that I'm showing you here is slightly atypical, but you can see that there are here a number of wells in which there's green, so there's GAG produced intracellularly, but no detectable GAG in supernatant, and also not any spliced, multiply spliced TAT-REV. However, there are also cells, and this is typical, in which both GAG and TAT-REV could be detected, and yet still there's no cell-free RNA detectable in supernatant. Those results were compared with the QVOA, which was done in the assay format shown here. The endpoint used here was HIV RNA rather than P24. And to jump to our most significant, I think, unexpected result, we found that using the cell-free supernatant HIV RNA gave results that were very similar to QVOA. So the correlation coefficient was quite good. And in fact, if you look on a sample-by-sample basis, in most of these samples, the discrepancy between QVOA and this inducible cell RNA measurement was within the range that is dictated just by the precision limit of the QVOA assay itself. So these were essentially statistically indistinguishable in most of those cases and quite well correlated overall. In contrast, cell-associated HIV RNA was not significantly correlated. Now, in a larger cohort, I think it probably would be. The correlation coefficient here was 0.3, but it was not statistically significant in this group. And it was, in general, more cell-associated HIV RNA. There were more cells that could be activated to produce intracellular RNA than extracellular GAG, by a factor of about 40 when we looked at GAG and a factor of about 6 on average when we looked at TAT-REV. This now shows you the same data compared with HIV DNA, and these cell-associated HIV RNA induced by activation was no better than DNA at predicting the QVOA assay. So all of the effort that we went to try to find something that was closer to QVOA, for cell-associated RNA in this study, we got no benefit from that. But again, the iCare, this is the induced RNA assay, looking at supernatant gives us a measurement that looks much more like QVOA. So this puts together the measurements that I've described. We also looked here at looking at the QVOA at a couple different time periods. Some people have propagated for different periods of time. And again, the bottom line is that the QVOA versus the cell-free induced RNA in the dilution assay format give very similar results within a factor of 2 and strongly correlated. In this study, whether for both of the cell-associated RNA targets we looked at, there was not a significant correlation. And we did find, as had been reported by the Mellers group and by Nicolas Chaumon's group, that induced cell-associated RNA was between DNA and QVOA values, but it was more correlated with DNA, or significantly correlated with DNA, but not with QVOA. In order to be able to demonstrate these correlations, it was necessary to do quite a number of replicates. And the data that I've shown you was analyzed based on the usual assumption of Poisson single-hit kinetics. But an additional benefit of doing so many replicates is that we actually can add in another layer of statistics in which we initially compute the estimated IUPM value or induced cell number value, and then go back and look for exceptions to the single-hit Poisson kinetics model and screen out potentially problematic wells. So for both the cell-free and cell-associated, in order to be able to measure the correlation. Well, first, the cell-free, in order to be able to measure the correlation we described, and based on our analysis for either one of these, in order to measure a 4-fold drop reliably, we think we needed at least 6 replicates, maybe not 12. That is more than has typically been used in QVOA assays in the past. So in summary, our study found that induced cell-free HIV RNA was a good surrogate marker for QVOA, and this suggests that the vast majority of the virus that is produced in a single round assay is in fact replication competent. So there may be even simpler ways to measure this. Although it was the case that we were able to measure induced cell-associated RNA much more easily than QVOA, it was not the induced cell-associated RNA was not predictive of QVOA. I think further sequence analysis will probably be necessary to tease out exactly what's going on. There's clearly variability between patients. I didn't show you all of the individual patients that we looked at, but there are significant differences in the amount of cell-free versus cell-associated. And in terms of the GAG versus the unspliced versus spliced RNA intracellularly, there are differences not only between patients, but also depending on whether we have frozen and thawed the cells or used fresh cells. And finally, based on our results, we think that dilution assay formats with a minimum of 6 replicates would be necessary to demonstrate the results that we've shown here and presumably to measure changes in cure trials. So with that, I'll be happy to take any questions.
Okay, one quick question. Otherwise, maybe you can ask during the break. That would be helpful. If not, then we'll move on. Thank you very much.